This is the beginning of part 3, which will conclude this tutorial. The goal for test 2 is to find and classify the tracks across scans and individuals. So you can go again from 300,000 fiber tracks to the 20 to 50 reference tracks and fibers. For each fiber tract, you're going to identify a reference fiber that you're dealing with. And the goal here is to maximize the reference fiber similarity between scans and subjects. Conceptually, what you're going to do is you have a source of reference fibers, as illustrated by the orange rectangle, and then you're going to test to see how well that predicts the rest of the fibers you've labeled within that fiber tract, as illustrated by the green line. For test 2A, it is within scan that you're predicting. So you have a set of reference fibers for each individual, and you're predicting the fibers of the rest, and the measure here is the degree of similarity of all the rest of the fibers within that track, but you must account for 80% of the fibers in the whole brain. For test 2B, it's a within person between scan similarity, so that now you're going to predict for each individual their tract, and then you're going to predict their performance in a second scan within that set. In test 2C, this is a between subject scan, so we're going to use brain 1, the first person, as our base, and then predict where the fiber tracks are on person 2 and 3 within that set. So to give you some examples in terms of the clustering, you might, for example, use the geometric centers as a feature of the clusters that you're dealing with, and within that set, use some methods such as k-means clustering on that geometric distance to identify your grouping of the sets. The classification will find the mean of the geometric centers of the fiber bundle of brain 1, scan 1, and then in brain 1, scan 2, with the closest geometric centers within those sets. And you'll identify all the fibers which have similar feature properties. You might take a two-stage approach, starting with clustering of the fibers into their bundles, and then classification of those into the within subject clusters of the subject that you're dealing with, and then between subject clustering of the set that you're trying to match. Again, in terms of the examples of the geometric clustering. Okay. In terms of task 2, the classification, this is just an illustration. In the between subject case, we first want to identify within scan what are the clusters that we're dealing with. Then we're going to do this making a mapping between scans, between brain 1 on scan 1 and scan 2, and brain 1 on scan 1 and brain 2 on scan 1. We encourage you to come up with your own effective approaches within that set. There are many possible approaches, and we hope contestant researchers will utilize multiple methods, particularly those that they're an expert at. The goal of the competition is to identify the best techniques that are out there. If you're an expert in a given technique, please apply it and include in your description how it worked. Some approaches may be better at some tasks than others. There is not necessarily an optimal approach for all the tasks. Include in your write-up what worked well and what did not. We encourage you to do sensitivity analysis of the components of your approach to identify what actually contributed to your successful entry in the competition. We provide a number of resources for you on the competition website, in particular a list of references and related fiber tract classification information, software to download and look at the data, useful routines in MATLAB, including routines to read and write the fibers. Some examples, there's a tool which will identify all the fibers that pass through some volume of union or intersection. So you can do the and not and or operations that I illustrated that our expert had done under computer control. Something to score your data on Brain 1, so you can do your testing on Brain 1 data. Something to show tracks to give you a brief visualization of the methods that you're dealing with. We will also be releasing next week some sample test program for each of the competitions. The goal here is just to give you an illustration of the reading in and writing out of the files, the scoring, the generating of the submission, and then you would have a place where you can put your own code for the techniques that you're operating with. We encourage you to add your expertise to the goal. This is a community effort to map the cables of the brain. There will be links to useful papers and programs, and if you have ones that you think are useful, please provide them. Suggestions for useful tools, subroutine code that might be used by others, and viewing tools that might examine the data. We're hoping to help build an open source library for the competition, and we will ask entrants to encourage to upload the tools. And after the competition, we will make those tools available, because this is a continuing process. So this ends the second tutorial. I encourage you to go to the braincompetition.org website for details and follow the details within that set. This is an important and interesting challenge to the data mining community to provide a method to map the cables of the human brain. Thank you.